Welcome, everybody. Hi, everyone. Thank you so much for joining us today at this symposium. My name is Dr. Samantha Loy, and I'm chair of the Australian Young-Onset Dementia Special Interest Group. This special interest group is a network of nearly 100 researchers, clinicians, and people living with dementia and families living with younger onset dementia. And our shared goal is of improving outcomes for people with young onset dementia. I would like to begin by acknowledging the traditional custodians of the land on which we gather here today. In my case, this is the Wurundjeri people of the Kulin Nation. And I'd like to pay my respects to their elders, past, present, and emerging. I extend that respect to Aboriginal and Torres Strait Islander people who are here today. I hope that you've all really enjoyed the Australian Dementia Forum over the past few days. In this symposium, we are going to focus on young people living with dementia and their families. We know that there are more than 29,000 people in Australia living with younger onset dementia. In the symposium, we seek to bring together experts to learn about the latest in young onset dementia and care. Today's symposium will run for about 2 hours and will be recorded and available to watch later on at a later date. And we'll hear from 3 incredible plenary speakers, and there'll be opportunity to ask questions of our speakers after the talks. I'll be joined by my co-facilitators, Dr. Adrian Withall and Dr. Monica Cations. We have a large audience today, and we appreciate keeping your cameras yourself muted and turning off your videos. If you have any questions for our plenaries, please add them in the Q&A box, and we'll try and get to them as soon as possible. So without further ado, I'd like to introduce our first plenary speaker. This is Professor Gil Rabinovici. He's the Edward Fine and Pearl Landreth Distinguished Professor in the University of California, San Francisco, Departments of Neurology, Radiology, and Biomedical Imaging. His work investigates how structural, functional, and molecular brain imaging techniques can be used to improve diagnostic accuracy in dementia and to study the biology of neurodegenerative diseases with the goal of accelerating treatment development. So I'd like to present Professor Gil.
Thank you, Samantha. Can you hear me okay and see my slides?
Yes.
All right, great. Well, really a privilege to join you here from San Francisco, California. It's late afternoon on May 31st, so we're still in May of 2022. I hope June is better. I'm going to be talking about how molecular imaging can help us understand disease mechanisms and heterogeneity in early onset and atypical forms of Alzheimer's disease. And when we talk about heterogeneity in Alzheimer's, you really can think about different dimensions. So one, of course, is heterogeneity in age at onset and genetic background, and that is really the focus of today's symposium. So we know that approximately 5% of all patients with Alzheimer's disease develop symptoms before age 65. And that's typically the threshold that's used to define early onset or young onset AD. Now, contrary to the belief that most of early onset AD is autosomal dominant forms of the disease, actually probably only about 5 to 10% of all people who develop symptoms at a young age carry a known mutation in APP or presenilin 1 or 2. And so the vast majority of patients who develop an early onset form of the disease develop so-called sporadic AD in the absence of a known autosomal dominant mutation. When we talk about heterogeneity, we can also consider clinical and anatomic phenotype of Alzheimer's disease. So while it's true that the majority of patients develop a memory predominant syndrome that affects the hippocampus and the medial temporal lobe circuits, a significant minority of patients with Alzheimer's disease present with primary language disturbance, a condition known as a logopenic variant of primary progressive aphasia, or a primary visuospatial and visuoperceptual syndrome known as posterior cortical atrophy, or more rarely a primary behavioral or dysexecutive syndrome that can resemble behavioral variant frontotemporal dementia. And there's overlap between these two dimensions of heterogeneity in that patients with younger onset disease are more likely to have one of these atypical or non-amnestic phenotypes. And so in this short talk, I'll talk about the different phenotypes of AD and how amyloid and tau PET have helped us to understand their relationship with patterns of amyloid beta and tau. I'll talk about modifiers of the phenotype, primarily age of onset and APOE genotype. And then in the second part of the talk, I'll describe the longitudinal early onset AD study or LEAD study. This is a prospective observational longitudinal multi-site study in the United States that is specifically focused on young onset forms of Alzheimer's disease that are not autosomal dominant, not covered by the Diane study. And I'll describe specifically PET results from the LEAD study. And so actually, the first reports of the patterns of amyloid PETs in patients with atypical forms of AD were reported in this paper that came from the ABLE study in Australia. So I'm sure you'll recognize all the authors. This study was published in Archives of Neurology in 2007 and described really a case series of patients with a primary progressive aphasia due to Alzheimer's disease who showed asymmetric uptake in the left hemisphere of PIB, the amyloid compound, or patients who presented with posterior cortical atrophy and showed predominant occipital patterns of amyloid PET retention. Now, in subsequent years, it became clear, and our group and others reported on this, that while individual cases might adhere to these specific patterns of amyloid PET, in general, at least at a group level, amyloid PET patterns tend to be very similar, regardless if patients have an amnestic form, a language predominant form, or a visuospatial predominant presentation of AD. And the patterns of amyloid PET really binding diffusely throughout the neocortex could not explain the very focal and different patterns of neurodegeneration that we see here with FDG PET. For example, asymmetric hypometabolism in the left hemisphere in patients with a language presentation, or posterior predominant hypometabolism in patients with PCA. There really was not a strong association between the pattern of amyloid deposition and the pattern of neurodegeneration. But as we started to perform our first tau PET scans, it became clear that the clinical phenotype is much more tightly related to the distribution of tau PET binding. So here are some of the first cases that we scanned with the 4-tau superior tau PET ligand in a patient with posterior cortical atrophy showing high retention in the occipital lobe, asymmetric left hemisphere retention in the lobopenic variant of PPA, and a temporal lobe and medial temporal predominant tau PET pattern in amnestic forms of Alzheimer's disease. And so just last year, we expanded on these findings. This is a study that was led by Renaud Lajoie in my group. And we looked at the association between patterns of amyloid and tau PET, clinical variability, and APOE genotype in roughly 120 patients that were followed in our Alzheimer's Center, including 69 patients with a typical amnestic form of AD, 21 with PCA, and 29 with LVPPA. You can see these patients in general had a relatively young mean age. There was no difference in the sex distribution, years of education, or handedness. There was a higher proportion of ApoE4 carriers in the amnestic subtype. I'll get back to that, though this was not statistically significant in this study. And overall, the groups were balanced in level of impairment as measured by the MMSE or CDR sum of boxes. So these are the average amyloid PET binding patterns as imaged by PIB in the amnestic, PCA, and LVPPA groups. And if these patterns look very similar to you, it's because they are. So on direct statistical comparison, really we found very small differences in the patterns of amyloid PET. The PCA patients had slightly higher binding in the occipital cortex. This is probably the most reproducible correlate of amyloid PET in PCA. The amnestic patients had slightly more amyloid PET binding in temporal lobe, but actually these differences were not significant after correcting for multiple comparisons. And in contrast, here are the average group level tau PET maps, showing again distinct patterns that are best appreciated again on the direct statistical comparisons, where we see a lot more tau PET binding in the language group in the left hemisphere language areas, as shown here in yellow, higher tau PET binding in the PCA group compared to other variants in the occipital cortex, and maybe a little harder to see on your screen, but higher medial temporal lobe binding in the amnestic group. So really the patterns of the clinical patterns and the neurodegeneration patterns very closely match the patterns of tau, but not so much the patterns of amyloid beta. Now, how do these different patterns of tau form? Rick Ossenkoppel, when he was a postdoc in the lab, used seed-based connectivity to compare patterns of tau PET in patients with MCI or AD to the normal architecture of the human brain as measured by functional connectivity or resting state fMRI. And so in this study, Rick used specific seeds in areas that are uniquely affected in different variants, like the right middle frontal gyrus for amnestic early onset AD, the right middle occipital gyrus for PCA, and the left superior temporal gyrus for LVPPA, as well as regions that are affected across variants, like the right medial temporal lobe and the right posterior cingulate. And what Rick found is that the covariance patterns of tau PET across patients very closely matched the patterns of normal functional connectivity in young healthy controls. You can see the great similarity between the tau PET patterns in the top row and the functional connectivity patterns in the bottom row. And so this really supports the idea that tau spreads through specific neural networks, perhaps via transsynaptic spread. But of course, what's more interesting than how heterogeneity in these patterns arises is why heterogeneity might arise. What are some of the factors that might drive tau to present itself in different brain areas? So this is a conceptual model that was introduced by Visha van der Fleer and Philip Sheltons in 2010 that said that patients with a typical amnestic form of Alzheimer's tend to present at later age, a mean age of about 75, and are more likely to be ApoE4 carriers. Whereas patients with these atypical non-amnestic syndromes tend to present at a young age, mean age in the mid to early 50s. And these patients are actually less likely to be E4 positive. They're typically E4 negative. And as we'll see, this conceptual model seems to hold very well in terms of the imaging data. And in fact, also matches another classification scheme that Melissa Murray proposed a year later that was based not on clinical features or neuroimaging, but on neuropathology, specifically the density of tau tangles. And what Melissa reported was that there was a hippocampal sparing form of AD where the density of tau was particularly high in the cortex. And this tended to be associated with atypical syndromes, younger age of onset, and E4 negativity. And in contrast, a limbic predominant form of AD, where most tau density was in the medial temporal lobe, tended to occur in older patients and in E4 positive individuals. So as you may have guessed by now, one way that ApoE4 might modify the phenotype in Alzheimer's disease is by changing the distribution of tau. This is data from the Lajoie paper that I introduced earlier, looking across the brain at differences between ApoE4 carriers and non-carriers, all with symptomatic forms of AD. And what Renaud found was that there was higher tau uptake in the medial temporal lobe, very specifically in the medial temporal lobe, in the E4 carriers compared to the non-carriers. And here you see the quantification. This has also been replicated by the Montreal group in ADNI and in the Triad cohort in Montreal. So this tends to be a very robust finding across different cohorts. ApoE4 drives tau into the medial temporal lobe associated with an amnestic phenotype. Now, age of onset affects both the overall burden and the distribution of tau, but not amyloid PET. So here you see the association in our study between age of PET and global cortical PIB, really no significant association, but a very strong association between global cortical tau and age, where the younger the patient, the higher the overall tau burden. And age modifies not just the overall burden of tau, but also the spatial distribution of tau. I'll show you that in a moment. Interestingly, this negative association between age and overall cortical tau PET was robust across all syndromes, amnestic AD, PC, and LVPPA. This was a very strong effect. And again, shifted the distribution of tau into the neocortex. So this is a voxel-wise correlation showing that younger age is associated specifically with higher tau PET uptake in the lateral temporal, posterior cingulate, parietal and dorsolateral prefrontal cortex, but not in medial temporal lobe or anterior temporal lobe. So younger age, shifting tau into the neocortex, and then patients more likely to present with these atypical non-amnestic variants. Now, so far we've talked about a clinical syndrome and associated it with neuroimaging findings. In this very nice study by Jake Vogel and Oscar Hansen, they actually turned this approach upside down. They looked at a very large data set of 1,100 tau PET scans from 5 distinct cohorts and applied a machine learning algorithm called SUSTAIN. This is an algorithm that can take cross-sectional data and actually derive those spatial and temporal patterns from the cross-sectional data. And what they found was 4 distinct tau PET patterns: a limbic pattern, a medial temporal lobe sparing pattern, a posterior pattern, and a lateral temporal pattern. These different patterns were very robust to longitudinal change. They replicated them in an independent cohort studied with a completely different tau PET tracer. And importantly, none of these patterns were really predominant. All were expressed in roughly 20 to 30% of patients. And if these patterns look familiar, it's because they're very similar to the patterns based on clinical phenotype that I showed you earlier. So the limbic predominant pattern was associated with an amnestic phenotype, ApoE4 positivity, older age of onset. The posterior predominant pattern with visuospatial impairment looking very much like the PCA pattern, older age of onset, that doesn't quite fit with our rubric. The lateral temporal pattern with an asymmetric presentation, maybe a language presentation. And finally, the medial temporal lobe sparing pattern, E4 negativity, younger onset. And so this heterogeneity in tau PET is there when you look for it in different ways. And you can really imagine how we might use these patterns then to improve our precision medicine approach to prognostication and monitoring disease progression. So we think that young onset Alzheimer's disease is a really important population for research to focus on. And in 2018, we launched the LEAD study. This is a multi-site NIH-supported study across the US. The contact PI is Liana Apostolova at Indiana University, Maria Carrillo from the Alzheimer's Association, Brad Dickerson from MBH, and myself are co-PIs. And in this study, we are specifically enrolling 600 patients with sporadic early onset AD who are not fit for Diane, are not really represented in ADNI. We're acquiring longitudinal annual clinical cognitive MRI, amyloid and tau PET, CSF, plasma, and genomic data. And really the goal is to inform the future design of trials specifically focused on young onset AD. And in the Q&A, we can maybe talk about why this is such a compelling population for clinical trials. So everyone in the study receives an amyloid PET scan with florbetaben. Approximately 76% of our participants so far, based on a clinical diagnosis of early onset AD, have been amyloid PET positive. And about a quarter, 24%, are amyloid negative. And I think this highlights some of the diagnostic difficulty, especially in early stages of making the diagnosis of early onset AD. And so the patients that are amyloid positive are followed in a cohort of early onset AD. And fortunately, we were able to obtain additional funding to also follow the patients who are amyloid negative to try to understand what is driving their clinical dementia at a young age. These are some of the tau PET patterns. So compared to control, we see a typical temporoparietal and dorsolateral prefrontal pattern of tau PET in most patients, but we're also seeing some of these focal variants, the PCA pattern, the LVPPA pattern, and in some patients, even though they're young, we see impairment with relatively low levels of tau. So this is also a very interesting group. In our early onset non-AD, in those who have impairment but are amyloid negative, about 30 to 40% have totally normal appearing tau PET scans. These are SUVR images, and here are W maps just to show you statistical relevance. And then roughly 60 to 70% have some tau PET uptake, but it's not in a typical AD distribution, more in a frontotemporal distribution or affecting the anterior temporal lobes. You can see this is a statistically significant signal. And so we think that at least some of these patients who meet criteria for early onset AD but are amyloid negative probably have an FTD syndrome that's evolving, and we'll continue to report on that as we obtain more data. This is just looking at the relationship between tau PET and the temporal lobe on the x-axis and amyloid PET and the cortex measured with centiloids on the y-axis. And you can see the controls in gray, the early onset non-AD in blue, and the early onset AD in sort of a darker blue. And what you can see is that the relationship between amyloid and tau is linear at this early stage of amyloid and tau accumulation. But then over time, tau PET continues to increase whereas amyloid PET seems to hit a relative plateau. So there's really no association between amyloid and tau at these later stages. And you can see that not all, but most of our early onset AD patients are really on this flat part of the sigmoid curve. And so this may have implications, for example, suggesting that even with mild impairment, patients with early onset AD may be less responsive to the clinical benefits of amyloid lowering therapies. Sorry, my light went out. I'm too bradykinetic here. Okay, just a couple more slides. We have longitudinal data for amyloid PET and tau PET in about 100 patients. Some of them have 3 time points. So not going to talk much about these spaghetti plots, but just to say that one of the main goals of the lead study is to compare this young onset population to late onset AD as measured in ADNI and in other data sets, possibly in ABLE, and really look at differences in clinical presentation, baseline and longitudinal change in biomarkers like amyloid and tau PET. And so just to summarize, a few take-home points. AD is a clinically and anatomically heterogeneous disorder, particularly when it presents at a younger age. The clinical features and neurodegenerative patterns are much more strongly associated with patterns of tau than amyloid beta. I've shown you that age and ApoE modify the AD phenotype, with younger onset associated with more neocortical tau and non-amnestic features, whereas E4 is associated with medial temporal tau and episodic memory loss. There are many other modifiers of phenotype that I didn't have time to get into, such as biological sex, the presence of co-pathologies, so you can see some of these references. And really, through studies like LEADS, we're really aiming to identify additional genetic, neurodevelopmental, and environmental factors that might predispose people to develop AD at a young age and to have different clinical manifestations. So sporadic early onset AD is an incredibly important area of focus for future AD research. I think this population has a lot they can teach us about mechanisms that drive susceptibility, that explain why the disease expresses itself at a young age, and also maybe drive clinical heterogeneity. And so I'll end there. I'd like to thank my collaborators at UCSF on the LEAD study, Bill Jagus at Berkeley, and emphasize that we do have postdoc positions available in the lab. So if any of you are trainees or are interested in a postdoc, please shoot me an email after today's forum. Thank you very much. And I think I'll take questions later.
Thank you so much, Gil. That is just so fascinating to hear. And we might actually do your Q&A now, so that I know how late in the day and late in the evening it's getting over there, so that you can run away when we're done. So then we've got some great questions for you. So I might ask a question from Kaylis Roberts asking, given the high level of correlation between fMRI and tau PET scans, for those of us clinicians who cannot access tau PET scans, would an fMRI be worth performing as a proxy for those with suspected Alzheimer's disease?
I wish I could say yes, but even some of the major proponents of resting state fMRI, like Michael Gracious at Stanford, Bill Seeley at UCSF, and others, I think really think of resting state fMRI as a very valuable research tool that can help us identify group level results. But as a single subject diagnostic tool, it is just too noisy and the effects are too small. So even though I know Bill and many others have been interested in trying to develop fMRI as a diagnostic tool to help us delineate these different networks, it just is not quite robust enough. But FDG PET, which is much more accessible, of course, than amyloid and tau PET, does a very good job of identifying the networks that are hypometabolic in these different variants of AD. And so if you're looking for a functional test that correlates nicely with patterns of tau, FDG PET is actually something that's worth thinking about in diagnostically challenging cases. And of course, I didn't have time to discuss today, but probably the most exciting development in AD biomarkers in the last few years has been the increasingly robust data coming out of plasma biomarkers of A-beta and phospho-tau. And of course, ABLE has contributed to that literature, as have other studies. And so I think that clinically, I'm optimistic that within a year to 2 years, we will have access to some of these plasma tests in the clinic. And of course, that will be much more cost-effective than doing PET scans and less invasive than lumbar puncture.
Yeah, that's great. I'm confident about that too. And that would be a game changer, I think. I've got a question in the Q&A directly that was sent to me, which is, in lieu of the findings about tau and amyloid, with tau being more sensitive, do you think the amyloid PET will go out of fashion and tau imaging will become the next or the newest frontier in imaging?
It's a great question. You know, tau PET is great, and I showed you very nice data, but it actually is not that sensitive. So the data suggests when you compare tau PET scans to neuropathology, that visually you are identifying BROC 5 to 6 tau spread. Maybe with quantification, we can identify some cases of BROC 4 pathology, but actually BROC 3 or earlier is probably not detectable, at least with our current tau PET ligands. Now, maybe the second generation tau PET tracers may be more sensitive to these earlier BROC stages, but when you're thinking about early detection, tau PET is actually not very sensitive, and amyloid PET turns out to be far more sensitive in identifying preclinical AD or even an MCI, depending on how you define MCI. Somewhere between 40 to 60% of tau PET scans might be negative. Of course, we know the average BROC stage in MCI is about 3-4, so it makes sense that we're not that sensitive. So I think at the dementia stage, tau PET and amyloid PET are similarly sensitive, and tau PET provides much more interesting regional information about the disease. But when you're thinking about MCI or subjective impairment or preclinical detection, amyloid PET is actually more sensitive. So I don't think that amyloid PET will, maybe it'll be out of fashion, but I still think it will be useful. And I think the plasma markers also won't put imaging out of business, because while they're very good at detecting the presence or absence of pathology, they are not very good at detecting the overall amount of pathology. They're not very sensitive as longitudinal measures. And of course, all that regional information about where the tau is, which is so critical, can't be derived from a single plasma measurement. So I think in terms of disease staging, in terms of spatial information, the imaging is here to stay as well. So I'm not going to put my PET scanner up for sale on eBay quite yet.
Yeah, and I mean, it makes me think about how sometimes even when things are out of fashion or not so sexy, it doesn't mean they're not useful, right? One last question is about the use of the MMSE and the those kind of generalist cognitive screens in these kinds of trials, when the person who asked the question has mentioned that executive functioning is such an important deficit in young people with dementia, and I wonder if you have any comments about that.
No, it's true. The MMSE, of course, is very biased towards memory and orientation and doesn't do a good job of capturing executive dysfunction. There's only 1 point for visuospatial function. The MoCA probably does a better job as a screening tool of encompassing all of these different cognitive domains. And it's actually a challenge when you're trying to compare different phenotypes of AD, because the MMSE, even the MoCA, even the CDR tends to weight memory symptoms over some of the other symptoms. And so we've definitely had patients whose CDR is 0, but they actually are very impaired because of PCA. It just doesn't show up on the CDR. And so that's always a challenge. What is the best way to try to control for disease severity in these non-amnestic forms? If anyone has a good idea, let me know. We've tried combining the MMSE and CDR, and that seems to work a little bit better, but I'm sure there are other approaches that are more sophisticated.
Yeah, thank you for that perspective. All right, well, Professor Rabinovici, thank you so much for your time. I'm going to hand back to Samantha now to continue on with hosting, and just thank you very, very much for your insights. Absolutely delightful to have you with us today.
Okay, thank you.
Thanks very much, Monica and Professor. We're really grateful for your presentation. Amazing. So I'd now like to present, we have a recording from Professor Sebastian Crutch. He's a professor of neuropsychology at the Dementia Research Centre, UCL Queen Square. His research focuses on rare and young onset dementia, exploring topics including dementia-related visual impairment, the dementia, and the arts. He currently co-leads the Rare Dementia Support, which exists to provide care, support, and advice to people living with rarer dementias, and leads a 5-year ESRC NIHR research program evaluating the impact of support groups that bring together peer and professional expertise. So here's the presentation.
Thanks, Samantha. Can I check that everyone can see the right screen good and everyone can hear this hopefully. Go back to the beginning. All good?
Good morning. Thank you very much, Monica, for the invitation to speak today. My name is Seb Crutch. I'm one of the psychologists at the Dementia Research Centre in London at UCL, and also have the pleasure of co-leading the Rare Dementia Support Service, which I'd like to talk to you a little bit about today. Before I start, I should probably define a little bit what I mean by rare dementia. I'm obviously not proposing this is a medical or formal diagnostic term, but more a term of convenience. So of the approximately 850 or 900,000 people in the UK currently living with dementia, obviously we know that there are people living with a whole variety of different types of dementia, and of course also people living with young-onset dementia, which I believe you're focusing on today. And the Rare Dementia Support Service tends to work with those people essentially the name of whose dementia would not be amidst the one or two that most people in the general public tend to know, i.e. Alzheimer's disease and vascular dementia. But just to nuance that a little bit, of course, we realise that there's a whole spectrum of ages of onset. We work particularly with people with atypical phenotypes, so people living with posterior cortical atrophy, logopenic progressive aphasia, behavioural or dysexecutive Alzheimer's disease, certainly dementia with Lewy bodies, frontotemporal dementia, PPA. And of course, we also recognise that all of these different dementia phenotypes are part of a spectrum. So we hope that services like ours cater to and support not only people with rarer forms of dementia, but also have something to say and to the wide variety, the vast majority of people who live with non-memory or under-recognised symptoms of Alzheimer's disease. And apart from also working with people with atypical phenotypes and young onset presentations, we also have a particular interest in working with people with directly inherited autosomal dominantly inherited forms of Alzheimer's disease and frontotemporal dementia. So that's the broad umbrella of individuals we're working with. In terms of why the support service exists, essentially it will be experiences much like you, I'm sure yourselves have had, of meeting people who have one of these diagnoses, who themselves were labouring under the impression that dementia just affected memory in older people, and so who are asking us questions in our clinic, in our research settings. Am I the only one? Am I the only one with this? And from a desire for us to connect people together who live with these conditions, who may be having a whole variety of experiences, some of which are, or uncertainties, some of which are shown here, to say, no, you're not the only one. You're not the only one struggling with that particular unusual symptom. You're not the only one who's trying to grapple with why me and my family. And actually by bringing you together with others, not just with professionals who can tell you about your diagnosis, but by other people who have experience of living also with your diagnosis, that we feel that's where you can find the best forms of support. This all started in the context of the specialist cognitive disorders clinics set up initially by Martin Rosser and Nick Fox in Queen's Square. And the first frontotemporal dementia, as it was then, the PICS disease support group, started right back in 1993, 1994. So quite a long history and pedigree, fitting into the wider pedigree and history of neurological study and endeavour here in Queen's Square. I found this quote recently from Robert Louis Stevenson, no less, saying that Queen's Square seems to have been set apart for the humanities of life and the alleviation of all hard destinies. And I suppose that's a rather writerly way of saying that we're interested in Queen's Square, not just in the basic science and understanding the mechanisms and processes underlying these conditions, but also in what it means to live with them. So the Rare Dementia Support Service was founded in 2016, bringing together a set of individual support groups that we had founded at that point, not just into FTD, as I mentioned, but also into PCA, PPA, familial Alzheimer's disease, familial FTD. And since we've added groups interested in focusing on YOAD, young-onset Alzheimer's disease, and also Lewy body dementia. And broadly speaking, RDS tries to offer specialist social, emotional and practical support services to people living with or affected by a rare dementia diagnosis. And I guess importantly, our group started out to try and support those people we knew directly or were giving a diagnosis to within our own clinics. but soon it became very apparent that there were relatively limited provisions for people living with these conditions elsewhere in the UK. So quickly the groups opened up to be open to any and all. So we received both referrals from fellow healthcare professionals, but also from by self-referral. People can get in touch with us, having found us on the website. The broad 3 pillars of the support service that we try to offer certainly involves support, as I just mentioned, trying to be holistic support, and support which is not trying to do everything. We don't offer case management or advocacy, direct advocacy for people. We try to be focused and have a remit around the specialist information and advice we can provide relating to people's particular condition, and then to work in partnership with local services in order to find the relevant activities and local experts and specialists who might be able to follow up on those care and support recommendations. We're also consider it very much our remit to be involved in education, and that means helping our members themselves educate those around them, as I'm sure you will have found also people being by far and away the most powerful advocates and explainers of their condition, particularly when given the right tools and support. And many of people, one of their common complaints coming to our support groups, one of their common complaints is that they feel they have to be both patient and doctor in so many settings. They're meeting people who quite understandably have never met anybody before with one of these rare dementias, and so they're having to provide that explanation. So they're an amazing conduit for us to pass on hopefully tailored and sculpted information delivered by particular professionals, be it speech therapists, optometrists, occupational therapists, to other people in their profession having had the experience of working with someone with one of these rare conditions. And of course, we're also interested in research. And by that, I don't just mean the type of observational and treatment-based work that the Dementia Research Centre has done previously, but increasingly we're trying to turn a spotlight on these support services themselves and understand how we both design, develop and evaluate novel types of service for people with specific dementias at particular points in the development of their disease. So as I mentioned, we now have these 7 disease-focused groups, but we also have a whole variety of other services, including one-to-one support to individuals and families and small groups. The 7 groups still meet in large format, naturally, as webinars during the pandemic, but we just started meeting back in person, which has been a real treat. And also offering a whole set of regional groups. We currently have about 30 regional groups around the UK, and also have been developing a number of online groups. So it's really nice now for people to be able to have regular contact. So monthly peer groups, usually either people living with or care partners meeting separately, but with others with the same diagnosis. And of course, regular email support and other forms of communication in between. So just to give you a broad sense of where we are in terms of the scale of this work. So currently we have just over 4,000 members at any one time, and actually a much wider historical membership and wider sets of families and friends represented by those members. And it certainly also saw a huge increase in membership during the lockdown when so many local services were not available to people. So people coming to us to be connected with one another online. We hope this is a reasonably international offering, and obviously that's an attraction for me of coming to talk to you today, is to say we're very open and keen to work in partnership with any and all. As the RDS remit suggests, we don't see any reason really why there should be geographical boundaries around the services that are available to people. And the direct support team, led by my fantastic colleague Nikki Zimmerman, who a few of you may know through some international initiatives, she and her team currently provide about 1,500 support calls and 1,700 support emails per year to individual members and families. On the educational front, we developed a couple of MOOCs, massive open online courses, a few years ago, which have had a large number of learners. We're always keen to develop those sorts of resources and make them more and more tailored. I think we feel that we've perhaps created courses in the past which have tried to explain what are the rare dementias, and are now keen to be focused more on what do you do about them, how can we provide direct practical support in interactive video-based content. And I'll also mention a little bit later the RDS impact study, which has about 1,000 different types of forms of participation so far, which is trying to monitor the effectiveness of this service. I think the real growth area for us has been these online meetings, and I'll be talking about why in a moment, why that has been such a helpful way of reaching out across geographic distance, but also to keep people connected much further into their disease course. And I'll also be mentioning a couple of other initiatives that are particularly kind of on our mind and on our focus at the moment. In terms of who we're reaching, I think we do still have a bit of a bias towards people in England, but our website usage suggests that we're beginning to get engagement from some of the other British nations in the UK. And the international element to RDS is particularly important to us. So it's always been quite international. So over half of our website usage comes from outside the UK. But we've been particularly excited recently to work with Mary Pat Sullivan at Nipissing University in Canada, who's just launched a sister service called RDS Canada, supporting young Canadians and Canadians of any age living with one of these rarer or inherited forms of dementia. And that would be a particularly interesting partnership for us to see how the decisions made about how these structures are set up and governed will also be replicated in different contexts, and as they try and reach out in a very different situation to people in both cities, in urban Canada, but also in rural Canada as well. And we in the UK, in terms of our governance, try and dance essentially between university, hospital and charity. We have a fantastic charity called the National Brain Appeal who do all of our fundraising for us. So we're not a charity in our own right, but we're trying to blend together the very best of what the NHS, the university sector and the charitable sector can offer in order to help us try and remain a fairly fluid and autonomous service, able to make changes and decisions to innovate and offer new services in a flexible way. But part of that reach is we're very conscious that we're very biased, sadly, towards middle class membership. And I'll be talking in a moment about how difficult many people find it to get a diagnosis at all. And so to try and reach out more to underrepresented communities, we've also developed a partnership with the UCL Centre for Translation Studies to develop linguistically and culturally appropriate resources, video content, stories to try to make RDS more representative of the people who are actually living with rare forms of dementia. So just in the latter part of the talk, I thought it'd be helpful just to talk about a few elements of the research that we've developed to try and evaluate these services. And this is all under this the umbrella or banner of this impact study, the impact of multi-component support groups for those living with rare dementias, kindly funded by the Economic and Social Research Council and the National Institute for Health Research here in the UK, which broadly has 5 work streams, looking both to understand the cohort of people who use a service like RDS and how their individual and collective support needs develop over time, to consider some of the more theoretical elements around designing this kind of flexible support service, and to develop measures that are tailored to people with young onset and rarer forms of dementia, to develop a number of online interventions and small groups for in specific populations like FTD carers, and also to understand the cost of these sorts of services. Our current services run entirely on donations from members and a small number of grant-making bodies. But it'd be really interesting for us to actually quantify how much particularly these peer group-led services cost relative to more traditional one-to-one or individual-based services. One element of the research that we've endeavoured to use the time to reflect and consider within the research grant is essentially to develop a theory of change for rare dementia support. So a theory of change is essentially a comprehensive description and illustration of how and why a desired outcome or change is expected to happen in a particular context. I guess for us, rare dementia supports is slightly tricky to fit into that format in the sense that we're not just trying to achieve a single set of goals, but to deliver and build a community really, which involves people with lived experience, professionals and the RDS team themselves in developing an organisation where we can try and deliver some of the key principles which are outlined and highlighted here. So I thought what might be helpful is for me just to talk through 2 or 3 of these just as I finish, just as illustrations of some of the decisions and changes that we've tried to make over time. So one of the key principles for us is essentially for people joining RDS to feel a sense of connection and continuity. We're not just trying to deliver a one-off intervention or help them with a specific situation. We're trying to draw people into a longer term relationship so that when or if other problems or situations arise in due course, and that they have a common point, a common thread of support that's run through all the different experiences. And this type of approach has been influenced by members saying things such as written here, which I'll just read briefly. So finding my way through the disconnected health and social care services over the following 3 years after diagnosis was exhausting and included sourcing help from over 20 different organisations and contacting and developing relationships with over 60 personnel. When my husband was admitted to hospital in July 2018, we were dropped like a stone by the organisations in the community I had to start all over again. And RDS was the only organisation from which we were not discharged. And I put that quote there not to sort of pat ourselves on the back and say, didn't we do an amazing job? Because clearly, we, as I said earlier, our remit is not to do everything for people. The very fact that this lady was involved with us and yet still having this experience, I think highlights not that we should be resting on our laurels, but more we were fascinated by this idea of not being discharged and thinking a bit more about what that means and how that links into broader notions and ideas about the continuity of care. And I think in particular, one of our anxieties was that because our support groups had evolved naturally and organically out of our clinics, we were concerned that we tended to see people around the time of diagnosis and in the 2 or 3 or 4 years following, when people, for example, were taking active part in research studies and coming down to London and having scans and blood tests and neuropsychological batteries and so forth. And we're conscious that some people around the time clinically when they were being discharged back to local services because they couldn't travel to London anymore and couldn't potentially reach London to participate in studies, actually felt a bit like they'd been a little bit used, or at least that they'd been a bit deserted. And so we've been particularly focusing in the last couple of years on trying to develop services for people in the middle and later stages of their conditions, to try to sustain and make equal the offer across all the different elements and stages of their dementia. One of those offerings has been to develop this later stage program where care partners of people living with a rare dementia are invited along for a group of 8 sessions, taking place weekly, and trying to focus in on topics which people might have heard about a little bit, such as sensory stimulation, the importance of diet and so forth, but also some of the topics that people haven't had an open format in which to discuss uncertainties and questions, and where we can help people to prepare ahead of difficult decisions around transitions in care, for example, or around palliative care. So that's been one element of the service. And I guess another way in which we try to offer continuity is also, and try to live up to that member's suggestion that you're never discharged from RDS, is also in offering bereaved services for people. And that's involved both offering pre-bereavement grief and loss groups, so for people who are experiencing non-finite losses, so whilst people are living with their dementia, helping them to understand the losses in roles and identity and different elements of life, but also actually when members sadly do die of their dementia, making sure that we continue to support people's partners and family members. And this is something we've been doing back since 2017, but which has really taken off, I think, with the advent of, or the familiarity of online services around March 2020. And so far, we're seeing that not only offering these support services online, meaning that more people are attending, and that we're reaching out to people on average who are living more than twice as far from our central base in London as they used to be, but also trying to explore, using psycholinguistic variables, the impact of these of meeting together on the kind of conversation that these bereaved carers are having with each other, which seems to show an increasing authenticity, which is a psycholinguistic variable that really means your ability to speak freely and authentically about your experience, and also a change in the level of the emotional affect that people are demonstrating as they become more familiar with living alongside and walking alongside their grief. Another element that we're particularly keen to understand is what is the nature of empathy and understanding that people find when they are able to be in peer conversations and groups with one another. So as I say, we've been running these groups for a number of years, particularly developing these smaller themed groups around later stage care, or in the case of this small pilot research study, into the set of groups we run around independence and identity, trying to get to slightly more abstract concepts of the impact of dementia and care upon individuals. And so this was care partners meeting together to discuss topics either directly related to their own independence and identity, or in the third session, the independence and identity of the person they were caring for, and trying to apply a number of recording these meetings, transcribing them, and using this Luke linguistic inquiry and word count software just as one of the tools to try and understand how do those conversations develop over time, and are these peer conversations, which can be somewhat demeaned by others. Some people view them as being sort of non-specific, fuzzy help, fun relationships, but not really a form of healthcare. Actually, to try and say, well, these are quite specific conversations. So, for example, the nature of the types of words being used, the degree of authenticity or analytical thinking really does not only vary between sessions according to the theme and trend of the session, but also seems to be replicable across a number of different groups. So this is just one pilot set of data, but we're trying to explore more and more of our services in this way. And finally, just to finish, I talked a little bit earlier about accessibility of our services, and trying to say, what can we do to make sure that we are as open as we can be to any and all living with a dementia, but also proactively reaching out to underrepresented communities. The map on the left is where we have members at the moment, but we're keen to recognise that there's sadly a very still strong relationship between people's socioeconomic status, as shown in this paper by Jitdal and Charlie Marshall and colleagues in East London, who found that those in the lowest deciles for socioeconomic status were much more likely to have a dementia unspecified diagnosis, much less likely to have one of the specific diagnoses that we've been mainly talking about today, that enables you to access care, or for other people around you to recognise that you might need non-generic dementia support, something much more tailored to your particular set of difficulties. And so we've got a number of initiatives with a equality, diversity and inclusion working group. We've started gathering this data for ourselves so we can monitor progress and change in our membership over time, and particularly just piloting a scheme called Champions and Cascaders, where members from a variety of backgrounds are being trained up by one of our fantastic support workers, Trish, to go out to local community groups in their area and engage people in the topics that are important to them, and try to broaden the dementia debate and discussion beyond memory into some of the other areas of cognition, such as language and social cognition and perception, that are particularly relevant to them and their experience of a dementia. So thank you very much for your time today. Just to briefly summarise, that I do think people living with atypical inherited young onset dementias have really specific and additional challenges and support needs that tend not to be recognised by a number of more generic dementia services. I do feel that, obviously massively biased, but I do feel that a blended peer and professional services which really recognise expertise in all its different forms have a critical role to play here. I think these people living with these rare conditions also really challenge so many aspects of the way we've set up our healthcare system, as they struggle to find this specialist tailored and continuous support. And that the community characteristics of these individuals mean that we really do have to go above and beyond to collaborate. So not just at the local, regional, national level, and of course across multidisciplinary boundaries, but also thinking about how we draw in other partners, both nationally and internationally, to try and deliver and share services when these people, where there's so limited resource available to these people, we've got to be sharing practice. So we would love it if you have elements of your service support group practices that we could learn from and perhaps link in with, that would be an absolute delight for us. So I hope you have a great meeting. I'm really sorry I'm not able to join you for a live Q&A. Here's a little bit more information about our support services, and thanks very much indeed for your time listening today.
Thanks. Unfortunately, Sebastian could not join us live for the Q&A, but if there are any questions, please put them in the Q&A box so we can send them off to him. So now I'd like to present our last plenary speaker who is live. It's Professor Muireann Irish, who is a cognitive neuroscientist at the Frontier Research Group in the School of Psychology and Brain and Mind Centre at the University of Sydney. Professor Irish's research focus is on the cognitive neuroscience of dementia. Using novel experimental tasks and multimodal imaging, she seeks to understand how complex feats of cognition are affected in dementia and to develop new ways of supporting those who are affected. So I'll hand over to Professor Irish. Thanks very much.
Thanks so much, Samantha. Can everybody hear me okay? See the slides okay? Great. Thank you so much for inviting me to present today, just to give an overview of the way some of our research is moving and how we're starting to think about some of the symptoms that are present in frontotemporal dementia and how we might better understand the emergence of some of these symptoms and try to develop better ways to help people live a more, I guess, fulfilling and engaged life when they do have these rare dementia syndromes. So this work has been conducted at Frontier, which is the specialist frontotemporal dementia group at the Brain and Mind Centre at the University of Sydney. And a lot of the work that I'm going to present today was championed by a PhD student in our group called Siobhan Shaw, who's been really pioneering some of this work. And we're very excited by the direction that this is going in. So today I'm going to focus on this idea of reward processing, which is closely linked to motivation. And so if we step back from the dementias just for a moment and think about our own proclivity to engage in certain behaviors and certain hobbies, even certain types of food, much of our behaviors are actually driven by this desire to sort of seek out and allocate effort to derive pleasure. And so many of the things that we do are motivated either by primary rewards, such as food or sex. These are more biologically driven, but there are also secondary rewards as well, such as music, seeking out art or beautiful, scenery. Money falls under this category as well. And when we engage in goal-directed behavior and allocate effort to secure a reward, what we know from fMRI studies of healthy individuals is that this experience of reward and pleasure sort of stimulates hotspots, hedonic hotspots in the brain. And this is particularly relevant in the context of some of the dementias that we study, because this reward circuitry in the brain targets many of the regions that we know are vulnerable from very early in some of our younger onset dementia syndromes. And so this is the brain's reward circuit here. It's centered on prefrontal regions in the ventromedial and dorsomedial prefrontal cortices. We also know that the anterior cingulate is very much involved, as well as regions within the striatum, such as the nucleus accumbens and also the pallidum and the putamen. And these regions are consistently implicated in neuropsychiatric syndromes in which we see alterations in motivation and reward processing. And many of these include depression, bipolar disorder, and some of the negative symptoms that we see in schizophrenia. And so all of this is very interesting from a sort of theoretical perspective. But when we think about some of the syndromes that we see in the clinic, where we're studying particularly frontotemporal dementia syndromes, it becomes actually incumbent on us to understand how reward processing is affected in these syndromes, because we're starting to gravitate towards a view in which we believe that many of the primary behaviors and features that we see in these patients are actually potentially driven by an underlying impairment or change in the way that rewards are processed and experienced by individuals living with these syndromes. And so we know under the heading of frontotemporal dementia, there are three main clinical phenotypes. And these include the behavioral variant of frontotemporal dementia, which shows a sort of constellation of behavioral changes. There is the semantic dementia, or the semantic variant of primary progressive aphasia, which is characterized more by a loss of semantic knowledge alongside some other behavioral changes as well. And then there's progressive non-fluent aphasia, which is much more a motor speech and effortful speech and production syndrome. And so in this talk, I'm going to focus on the two syndromes here, so the behavioral variant of FTD and somatic dementia. And so This is just giving a very sort of brief overview of some of the clinical features that we might see when an individual with behavioral variant frontotemporal dementia presents to the clinic. Now, these features will present in different ways, and they're not necessarily characteristic of all patients that we see, but in general, we will tend to see diminished empathy and capacity to engage in social cognition. And we see apathy or loss of drive, changes in the ability to regulate one's emotions, insensitivity to loss. And so we often see changes in financial capacity and decision making, as well as changes in eating, sometimes inappropriate behavior and disinhibition. In somatic dementia, we also see some of these changes in emotion processing, eating, as well as the loss of empathy, but it's much more likely to be driven by this impaired comprehension and loss of conceptual knowledge of the world around them, as well as patients who have more right-sided predominant form of somatic dementia might show changes in facial recognition, as well as some changes in behavior, being more likely to engage in rituals and stereotyped behavior. So why is this relevant then in the context of reward processing? And so this is a question that many researchers in the frontier group as well as overseas have been asking over the last few years. And if we think about the many features and changes that we see in people who present with frontotemporal dementia, it becomes sort of evident that many of these changes can actually be potentially explained by a diminished capacity to experience rewards. And this is evident across a whole host of different features in terms of eating behavior and food preferences, this loss of interest in hobbies and pursuits that were formerly pleasurable to the individual, social withdrawal and disengagement from social scenarios, loss of intimacy, and as we'll see, changes in sexual behavior, as well as this impaired capacity sometimes to make financial decisions, which largely stems from insensitivity to loss. And so we've been thinking about these many seemingly disparate symptoms and wondering if an underlying deficit in reward processing might actually be a more parsimonious way of understanding these sometimes variable presenting symptoms. And so work that's been pioneered by Rebecca Ahmed at the Frontier Clinic has demonstrated that eating changes in frontotemporal dementia are actually highly prevalent and actually quite pronounced in the behavioral variant and somatic dementia. And so Rebecca devised a very clever sort of ad libitum eating task where participants weren't aware that they were going to be assessed in terms of what they were taking in. And so following their fasted blood collection, they were then allowed to eat as much of a particular type of food out of an array of different breakfast foods. And what Rebecca noticed following weighing all of the different dishes, that the caloric intake in the behavioral variant group was significantly higher than that of other forms of dementia. And she followed this up with a separate study where she varied the sugar, how sugar laden and eaten mess dessert was that she had made. And she asked participants to rate the different desserts, which varied considerably in terms of their sugar sort of loading, and found that behavioral variant FTD, and somatic dementia patients showed a preference for the highly sugar-laden foods, whereas patients, people who presented with Alzheimer's disease and healthy older adults really disliked this sugar-dense sort of food. So there are clear changes in eating behavior, and that we have shown that these actually relate to structural changes in the brain in many of these key reward processing regions. At the same time, we also see a sort of loss of intimacy and a loss of sexual drive in many of our dementia patients. And so this seems to run counter to this idea that, you know, patients are hypersexual. In fact, we find the complete converse profile. Many of our carers are reporting that the people who are living with Alzheimer's, with behavioral variant FTD and somatic dementia, instead are showing evidence of hyposexuality, so a diminished drive and a diminished sort of seeking out of those intimate behaviors that used to be characteristic of the of that pair. And this again is related to key regions in that reward processing circuit that I presented earlier, including the caudate nucleus, suggesting again there's something to do with changes in this reward processing network that are perhaps driving some of these diminished or dampened responses to pleasurable activities. And we also see a very pronounced loss of drive. And so this is characterized by apathy, which is really this inability to initiate and to engage in goal-directed behavior. And so work that we've been doing with Fiona Comfer and Stephanie Wong has shown that this is highly prevalent across many of the dementia syndromes that we see in the clinic. It's highly predictive of functional deterioration, of increased levels of stress and burden in the care partner, and as well as it's predictive of that entry into residential care settings and patient mortality. So it's really important to understand how this loss of drive manifests and how it actually predicts functional outcomes. We know as well that through a lot of separate studies that we've done, that again, we can chase or sort of trace these emergence of different forms of apathy. So it's not just a unified construct, it can vary across several dimensions, including affective, behavioral, and cognitive. And so these profiles of different dimensions of apathy are actually really important to understand, because we can see different profiles depending on the subtype of dementia, as well as a lot of individual variability, which is crucial to understand in the context of trying to deliver targeted interventions to the individual. But one of the areas that we have found that had been neglected within the literature was actually the idea of the experience of pleasure. And as I alluded to earlier, this falls very heavily under that reward processing rubric. And so we know that in many neuropsychiatric syndromes, loss of pleasure is actually a characteristic and very early feature where there is a decreased interest in, but also a decreased sensitivity to formally rewarding experiences. So the things that we typically enjoy and seek out and allocate effort to in our daily lives seem to lose their sort of rewarding flavor. And so people no longer enjoy, for example, a beautiful sunset, or enjoy the taste of a coffee or the smell of freshly baked bread. And so these kind of diminished sensitivity to formally pleasurable experiences is called anhedonia in this neuropsychiatric literature. And so we know that this stems from the ancient Greek for without pleasure. And it's simply put is a compromised capacity to experience pleasure. But it also refers to the diminished and compromised capacity to anticipate, to seek out, and to allocate effort to engage in pleasurable experiences. So there seems to be quite a considerable overlap in terms of perhaps apathetic behaviors. And as I've said, it's widely documented in many of these clinical disorders, including depression, schizophrenia, and bipolar disorder. But what we found was quite surprising was that no study, to our knowledge, had actually explored whether this capacity for pleasure was in fact altered or intact in frontotemporal dementia syndromes. And so we have good reason to believe that anhedonia would actually be a key feature of FTD, particularly in the behavioral variant, because many of these initial presenting features, the loss of drive, the loss of interest in former hobbies and pursuits, social withdrawal, loss of intimacy, disengagement, all of these actually might reflect a genuine loss of hedonic tone. And if that's the case, then it might actually be possible for us to intervene with targeted interventions to promote that sense of pleasure again, and actually in turn kickstart some of these more goal-directed and effortful behaviors. So that was the rationale for some of the work that we've been doing. We also were interested to see a growing literature of psychiatric and motivational disturbances in some of these syndromes. So as well as the fact that many of the imaging studies that we've been doing, as well as other research groups internationally, highlight that many of these regions in the reward circuit of the brain are actually affected in the FTD syndromes. We see striking prefrontal and striatal degeneration from very, very early on in the disease trajectory. So there's this whole clinical, behavioral, and neuroanatomical rationale for actually starting to understand and study anhedonia in FTD. And so in quite a large study that we published last year in the journal Brain, we recruited, it was 172 participants, and we used a very widely used measure of pleasure that's been used within the depression, anxiety, and schizophrenia literature called the SHAPS, which is the Snaith-Hamilton Pleasure Scale. But we wanted to modify this to ensure that we were capturing a reliable change from the pre-morbid behavior of the person with dementia. So we asked both the patients and the person caring for the individual to rate the individual's capacity to derive pleasure across a number of different scenarios, pre-diagnosis, so their pre-morbid capacity, as well as at the current moment. And this allowed us to understand their pre-morbid just personality and disposition, but also a change from that early pre-dementia phase. We also wanted to ensure that any changes in the capacity for pleasure did not simply just reflect depression, because anhedonia is a major feature of the depressive syndrome. We wanted to ensure that we were controlling for this. And we also assessed apathy using the dimensional apathy scale. And so we collected all of this data across a number of different subtypes of frontotemporal dementia. And they also underwent structural MRI through the Frontier Clinic, which enabled us to actually then pinpoint the neuroanatomical basis of any changes that we did observe in terms of the level of anhedonia that our people with dementia were experiencing. And so what we found, first of all, was that there were distinct profiles of anhedonia depending on the subtype of frontotemporal dementia. So in behavioral variant frontotemporal dementia here in red, and in somatic dementia, we saw significantly lower levels for the capacity to experience pleasure. So this means higher levels of anhedonia relative to a healthy older control group, but also a control group of Alzheimer's, individuals living with Alzheimer's disease. And interestingly as well, we found that despite having apathy, our PNFA patients, these are the progressive non-fluent aphasic patients, and our Alzheimer patients both showed clinically significant levels of apathy, which is the reduction in goal-directed behavior, but they didn't actually report having a diminished capacity for pleasure. So this provided sort of the first key or piece of the puzzle that perhaps we can dissociate between anhedonia and apathy. We also wanted to establish that any clinically significant anhedonia that we were capturing was a change from pre-diagnostic levels. And we did show this across the groups, that there was a significant change in how the patient and the carer had rated themselves pre-morbidly to the current moment. And then on the neural level, what was really interesting for us from a more therapeutic aspect is that anhedonia, apathy, and depression all showed distinct neuroanatomical signatures. So anhedonia was related to many of those regions that I showed you in the brain's reward circuit earlier on. So medial prefrontal and orbitofrontal cortex, insular regions, as well as the striatum. And there was only one region of overlap between anhedonia and apathy, and that was a very small cluster in the right prefrontal cortex and the orbitofrontal cortex. So this suggests to us that actually they have distinct neural signatures, which might make them amenable again to targeted neuropharmacological interventions. And so it suggests that maybe there's a way for us to start, pinpointing specific therapeutics that could be repurposed to target anhedonia or apathy, depending on the group that we're looking at. And finally, I think it's always, all of this work is very interesting. We're really excited by it, but it's important to bring it back to the clinical implications. So if we have, people with these dementia syndromes who are displaying a clinically and significantly attenuated or diminished capacity to experience pleasure, does that actually predict functional outcomes? And if so, then it would really suggest to us that there's a clear need for us to do something about anhedonia from very early on in the disease syndrome. And so we took a subset of very well-defined behavioral variant and Alzheimer's disease patients. And we looked again at their ratings on this pleasure scale, their depression ratings, as well as their motivational changes on the Cambridge Behavioral Inventory. And we wanted to see which of these measures was the best predictor of functional impairment on the frontotemporal dementia FRS, which is their functional rating scale. Because this would tell us then if anhedonia or if depression or if motivation are more likely to predict impairment over time. And so we specifically looked at the change in pre-morbid levels of pleasure as well as the current. So we wanted to see that drop from the patient's pre-morbid disposition to now, was that predictive? And what we found was really interesting. So in the behavioral variant frontotemporal dementia group, we found that the strongest and only actually significant predictor of functional impairment was that change in anhedonia from pre-morbid to current levels. So anhedonia and not apathy or depression was predicting functional decline in BVFTD. And then, which is remarkable, on the other hand, we actually found in our Alzheimer patients that it was the change in motivation. So this is higher levels of apathy were actually predicting functional decline in our Alzheimer group, and not anhedonia and not depression. So we can see here that we can have two different syndromes who are showing different changes, functional impairment in their daily lives, but actually the drivers of those changes are actually very distinct, arguably related, but also very distinct mechanisms. And so this is offering us some kind of hope that perhaps there's a way that we can now go in and target different aspects of these motivational changes to do something to actually enhance quality of life, to maybe stave off some of these functional impairments and improve well-being for the person living with dementia and their care partner. So all of this is to say that we're really excited by this work. We're really, we feel like we've stumbled upon a symptom that has not been looked at really in detail and warrants a lot of significant attention across our frontotemporal dementia groups. And we're now starting to look at how anhedonia might be a multidimensional construct and how different aspects of anhedonia might be affected. And this could range from being able to anticipate pleasure via anticipatory sort of mechanisms versus the more consummatory aspect where you have the reward and then you actually can derive pleasure from it in the immediate sense. We're also curious to start exploring how anhedonia might relate to a loss of future thinking or foresight in dementia, as we know that these are problems that are occurring in parallel, and we're trying to tease apart, which might be the driver, which is the cause, and which is the effect. And finally, we're starting to move ahead now to look at, can we develop, targeted interventions to improve anhedonia, because I believe this would actually have immense bearing on quality of life for both the person living with dementia and their care partners. So I will stop there, and just to say that this work has been really championed by Siobhan Shaw. We're very grateful to all of the participants and their families in our studies. They're just amazingly generous with their time. Of course, all my colleagues at Frontier and Forefront. And if you want to get in touch, please email. We'd love to hear your ideas, and we're very keen to collaborate as well. So thank you.
Fantastic. Thank you so much, Professor Irish. I think you can see the reactions coming up on the screen of such fascinating results. And I think we all in the community of young onset dementia research are really excited about this new frontier of research that you and your colleagues have uncovered, and really congratulations on all this fantastic work.
Thank you.
I have a few questions for you, and if anybody has any more that they'd like to add, please go ahead and pop them in the Q&A box. The first one is from one of the geriatricians working here in Adelaide, who mentions that apathy, in his experience, apathy seems to be more concerning to the spouse or the family or the carer than it actually is to the person living with dementia themselves. And wonder if you have any comment about that.
Yeah, so actually that is a really interesting point, because yesterday I went back to the data, and myself and Siobhan were trying to understand if we could find any relationship between anhedonia and carer distress or carer burden. And we didn't find any relationship between the two, but we do find a very strong relationship between apathy and carer rated stress and burden. So that would tie in very well with that geriatrician's observations. And I'm just wondering though, just because it's not overtly burdensome to the care partner, that doesn't necessarily mean that it isn't very troubling for the individual living with dementia. And I think I have a hunch that actually the anhedonia component is driving the apathetic behavior. So we need to start thinking about how we could actually measure that and demonstrate that sort of causal pathway. And I think if that's the case, then the anhedonia might be more amenable to treatment early in the disease course, and might actually have a knock-on effect of preventing highly, sort of inertia and that sort of withdrawal of behaviors and disengagement that is so troubling for the care partner.
Absolutely, and I think from a psychological perspective, that makes total sense that these apathetic behaviors become reinforced and become routinized through the via the mechanism of the anhedonia. And that is a question I've gotten directly in the chat about in terms of interventions. I know this is very early days, and just wondering if you have any comments about the potential for intervention, and whether you think that we're going to need to rely on drug interventions, or whether you think psychological interventions might have a role here.
Yeah, so I mean, it is very early, so I'm hoping to we can actually start a conversation, because this is something I really want to pursue. I think personally, I think we also need to understand that multidimensional aspect as well, because we can't just say that someone is anhedonic and then try and get them to engage or, enjoy something that they were never geared towards. So I think we need to do more work kind of charting very specific dimensions along which someone can be anhedonic, or along which pleasure might be attenuated, and also then design interventions that are worthy and sort of personalized to the individual. So there's no point trying to make someone like music more if that was never something that they were interested in the first place. So I think a lot of this will have to be tailored around personalized interventions. There is some evidence in, again, the neuropsychiatric literature that ketamine might be a good way to treat anhedonia. I mean, we don't know how that would work, and this repurposing of drug treatments from one population with very unique, underlying neurobiological mechanisms to another is not something that I think we should, rush into without fully understanding the underlying mechanisms that are at play. But certainly we're going to be looking at both aspects, starting to work on psychological interventions, because I think we have to do something, and also trying to better understand the neurobiology, so that maybe in the future that might be something that we could also look to do.
Yeah, absolutely. And you and I have chatted before about, I really have no idea where we go for this, but I wonder if we can learn things from other neurodiverse groups that experience anhedonia, groups like people with ADHD, people with autism, groups. Who knows? So much opportunity. One more question I have in the chat box is regarding the eating changes that you noted between different types of dementia. One of our attendees asked whether you've noticed any, or of any differences or changes in olfaction between different dementia types.
Yeah, so this is another a very interesting area. Now, this hasn't been the focus of the work that my group is doing, but there are a number of studies that suggest that perhaps that might be an early marker of neurodegeneration. And there is some literature out there to suggest that, and as well in the neuropsychiatric syndromes, there have been lots of reports of changes in olfaction. So I'm not sure, I'm not fully up to date on that literature, but I think all of these sort of sensory experiences are important to understand in that overall context, because they feed so directly into, trying to foster even the retrieval of memories, to try and structure the environment to be more, engaging and appetizing for the individual. So I think we have to understand across a sort of all of the senses, all of these sort of different activities, and we're trying to actually distill these various aspects into a better way of measuring anhedonia as well, that's more tailored to older adults. So most of these questionnaires are actually designed for younger people with depression and bipolar disorder, who are typically 40 years younger than many of the participants that we see. Many of the questions are not entirely suitable. So we're trying to take all of this information and think of a better way as well to go about assessing these very complex constructs.
Yeah, absolutely. All right, well, in the interest of time, I might wrap up our Q&A there, but Professor Irish, thank you so much for your presentation and for your time. I'm so generous with your time today. We really appreciate that. What I'm going to do now is hand over to Dr. Adrian Whittle from UNSW over in Sydney, and Adrian is going to lead our next part of our session, which is a panel session. So Adrian, I will hand over to you and ask all of our panel members to please pop on your videos and take it from there. Thanks.
Thanks, Monica. It is just such a pleasure to be here today, and such a pleasure to do this panel with such a diverse group of speakers. It's always been really important to me in my work that we hear from everybody and hear all the voices that should be in the room, particularly when it comes to, getting researchers together with health professionals, but also people who have, skin in the game, which are the people who are living with dementia, and also the people who are supporting people living with dementia. So thank you for having me in this forum. Now, I'd just like to welcome everybody that's here. I'm going to introduce one by one. So for starters, we've got Professor Olivier Piguet, who's an NHMRC Leadership Fellow and Professor of Clinical Neuropsychology at the University of Sydney. Good morning, Olivier.
Sorry, after all these years of all these months of COVID, I'm still struggling to unmute myself. Good morning, and thanks for having me. It's great to be part of this forum.
We will let you do your push-ups later for that one. Okay, and next I'd like to welcome Associate Professor Jade Cartwright. Now, Jade is a speech pathologist and also a researcher at Curtin University. Good morning, Jade.
Hi, thank you. Really delighted to be here as well. Thanks very much.
And the next person to give a warm welcome to is Judy Dimel, who's a nurse practitioner for Queen Elizabeth Hospital Neurology Service in Adelaide. Hi, Judy.
Hello, everyone. Thank you for the opportunity to be on the panel.
And the next person on my list, I'm going around in order here, is Isabelle Burke. Now, Isabelle is here as a dementia advocate and also a care partner. Hi, Isabelle.
Hey, everyone. Thanks for having me.
And last but by no means least is Bobby Redman, who has been on both sides of the fence, like me, who's been a psychologist and now a dementia advocate. So welcome, Bobby.
Good morning, everyone, and thanks for having me. Looking forward to it.
Okay, so we're with all the thumbs up and applause that we have going here, we're going to get this session started. So just to come back to where we started off, Olivier, my first question is actually for you. And I think what we would like to know is, what do you see as the strengths that we have in Australia around young onset dementia? So where are we particularly performing well in? But then where do you think are the fantastic opportunities that you have? Where do you think there should be investment and the development of more expertise?
That's such a good question, you know, and that forced me to reflect a little bit about what's been happening in Australia in the past 10, 15 years. And broadly speaking, I think we've made such progress in understanding these younger onset dementias presentations compared to even where we were 5 or 10 years ago. And I'm going to highlight a few points which I think are really important because of development in novel technologies, but also at the clinical level, and also in terms of intervention and clinical trials. I think in Australia, we are fortunate. We have a diverse group of outstanding researchers and clinicians working in the field. I think because of the where we are in Australia and being isolated physically from the rest of the world, that also has forced us to engage with other groups internationally. And I was looking at who's collaborating where, and Australians are very much involved in all the big consortia in younger onset dementias, as well as other aspects of dementia. So there's really a dynamic force of people in Australia that is extremely well connected in my mind. And also within Australia, we tend to be generous, friendly, and collegiate people who like to work together to find solutions, to find ways to make a difference to our understanding of these dementia syndromes, how we can diagnose them better, how we can improve the prediction of diagnosis, and then try to set up interventions. And often in a very clever way, just listening to the work that Marin is doing at the moment is an excellent example. You know, this curiosity and this interest in looking at these dementias in details to try to improve how we can identify people who are in that category, so improve the diagnosis, and then find ways to set up a system that will improve management over the long term. You know, just just to how long do you want me to talk for? Because I can speak for a very long time, but because I also want to address the second part of the question, where do I think the weaknesses are? But in terms of developments, recent development, I think what we know about the genetic forms of these disorders has changed the way we can tackle or understand the biological mechanism. So we now can identify genetic mutations, genetic abnormalities. We know of certain genes that we know will cause the disease, but we also now has the capacity to screen for many more genes and much more cheaply. So it means that there's the prospect of being able to have individualized genotype profile that will also inform us about some of the risks of developing some symptoms or the disease trajectory in people diagnosed with young onset dementia. So I think for me that's a major advance. And again in Australia we have people working on these genes, so the genetic development, so looking at the role of epigenetics and these very basic aspects of these disorders. There are people looking at cell lines, they're using stem cell and trying to create in vitro models of these disorders. And again, looking down at very basic mechanistic ways of understanding what is happening in the brain, what is happening at the cellular level, that will then help develop animal models. So they also have people working on rat models of dementia or mouse models of dementia, changing the genetic makeup of these animals to understand again how the disease starts and progresses and how we can modulate to change these mechanisms. And then we move on to the more clinical aspects, which is the general knowledge in the Australian researchers and clinicians, I think has improved tremendously in how we can diagnose people. And certainly, I think we are much better equipped at diagnosing dementia in general, but also different types of dementia, and certainly younger onset dementias. I think that the next challenge is to refine the differentiations between, because when you present in a clinic or when you go to your GP and you report changes in behavior, personality, memory, and in your 40s, the GP is not necessarily going to think dementia. Often it's going to be there's something with depression, there's in a more psychiatric space rather than neurological, neurodegenerative space. So there's the challenge in my mind is to improve the diagnosis at that level. How can we improve the diagnosis and distinction between neurodegenerative disorders and neuropsychiatric disorders?
And I think it's starting in that primary practice space, isn't it, too?
Yes,
it's sort of that primary care GP practice, like we've made a lot of movement with the specialists, but we need to take that step back to where people present.
That's right. And I think that leads me to the second part of the question. I think where we need to put more work, there are two aspects. One is, and someone has alluded to that in the chat room, is that the post-diagnostics care. We know a lot more about diagnosis, but then what do we do with this information? How can we, and that's what also Seb Crutch told us about, how can we set up a system that will help the person with the diagnosis, person with dementia, and their families go through this journey in a way that is meaningful and where they can find the information that they need, they can find the support that they need, where we can set up interventions and support groups in many different shapes or forms that will be useful at different levels of the journey. So early on post-diagnosis, but also later on when there are different aspects of the disease that will emerge and different questions that will need to be answered. So in terms of there's that aspect of support, and but also in terms of education. So educating GPs, as you mentioned, Adrian, I think is really important. Even educating specialists, I would argue that some people are very good, some people less so. And how do we address that? How do we disseminate this information to the wider medical communities, and not just medicals, but also allied health, and then set up a system where if you're not sure, you could then talk to someone in a specialized center who could then help you get to a diagnosis that is appropriate.
And I think, Olivia, I'm going to cut you off there, because I think that's a perfect segue into talking to Isabelle and Bobby, actually, because you're living with dementia, you know, you're experiencing everything in that real world. So I guess from people who've, you know, you're walking through that diagnosis space, you're walking through the care space as well. What do you two, with lived experience, and I might throw to Isabelle first and then Bobby, what do you see as priorities then for researchers? What can we do better to meet your needs?
Yeah, well, I'd echo the points that both yourself, Adrian, and Olivier made about time. Well, for me, it's accurate and timely diagnosis. And I think particularly Marin's presentation and some other things we've heard this morning have highlighted why an accurate diagnosis is so important, because how we care for that person then changes as a result of which form of dementia they've been diagnosed with. But for me, it's looking at what are the early behavioural changes that are indicators of brain change, and how do we teach the medical professionals, as you say, the GPs, the specialists, to spot the early warning signs and then seek out an accurate diagnosis. In my mum's case, she was diagnosed with dementia, with depression and anxiety for 15 years. And then, you know, at the end of 15 years, we find out that actually it's something else. And I also think that an emphasis should be placed on how to improve quality care practices. And as we've been saying, post-diagnostic care. But once we improve those care practices, people won't, well, we would hope, would no longer be so fearful of what's in the future for them. And that'll help destigmatize dementia and offer people a sense of hope. And that hope, I think, is really coming from elevating the voices of people living with dementia, hearing from people such as Bobby and her message and her life story, which is so inspiring. And that just shows people that your life's not over once you get a diagnosis, that you can live a rewarding life, you can give back to the community. But on the other hand, you know, my family's example was that my mum wasn't able to live well because she was diagnosed so late in the disease progression. And she wasn't able to engage in those activities that really you know, gave her a sense of purpose for so many years in her life. So I think a question I would put to researchers is, how do we creatively allow a person living with younger onset dementia to continue to engage in these things that have brought them fulfillment? And how do we continue to give people a sense of purpose, even once that point has passed where they're not able to engage in the same way as they used to? And I think that this is so important, particularly for younger onset dementia, is because you've essentially plucked someone out of their life at potentially the peak of their involvement in community, in their involvement in the workforce or their family. You know, my mum's, you know, my sister was 18 when mum was diagnosed, the youngest. So, you know, that's at when, you know, she's supposed to be entering into this new phase of life. And so how do we allow people to continue to do that? And it will have to be a really creative way, but I think there is a way of doing it. And yeah, and then also how we communicate the disease progression with families. That's something that I feel was really missed in my situation. I was 20 when my mum was diagnosed. I was 25 when she died. And I didn't know that those early warning signs of like difficulty speaking, I didn't realize that that would progress so quickly, and that would lead to me not being able to communicate to my mum. And you know, for someone like me who was so young, there were so many questions I wanted to ask her, but because I wasn't given that information at the start, that this is the harsh reality, that you know, within a few years, it's very likely that your mum won't be able to speak. I think we sort of rob people of the ability to face those harsh realities, because whether we're trying to protect people from it, but you know, information is key, and the more information we give people at the point of diagnosis, the better prepared they are as they enter into the next few stages.
And Isabelle, I think that's a really important point, because you know, I think it's always good to give people information. They don't have to read it, they don't have to be ready now, but at least if it's there and they have the ability that when they're ready, they can engage with it. People who used to come to our memory clinic who were younger, and it was one of the clinics that we had where people could come in through aged care at 45 or 50, they would say, please don't give it to us all in that one diagnosis session because it's overwhelming right now, we can't take it in. But we would always do a follow-up session, which was really nice. But unfortunately, that service doesn't exist anymore because it was deemed, you know, not very viable for funding. But I think that's really important, is to enable people at least to have that choice about what they do with the information and the knowledge. Yeah. And Bobby, passing over to you, how does that resonate for you, and what points would you like to bring up on that issue?
Absolutely. Yeah, look, first of all, I'd like to go back to what Olivia was saying. And yeah, I mean, diagnosis is the key for us. As mentioned, we tend, they don't want a diagnosis. Doctors don't want a diagnosis. They don't even consider dementia, really. Even, I mean, I was a psychologist, and you know, after a couple of years of symptoms, I recognized that things weren't right. But it's almost as if the doctors are in denial and really don't want to run the screeners that are available and look at everything. So, you know, getting that message across that really it's vital to be doing that. And as you say, it's not just the GPs, it's the specialists too. My neurologist was almost in the same boat. It's, you know, it's kind of, well, you know, you're doing okay. That was my diagnosis for, you know, several years. You're doing okay. It was kind of, you know, I knew I could see deterioration, but nobody wanted to acknowledge that things were going pear-shaped for me. So I think, you know, getting that message across to GPs, so the translation of research into, you know, transitioning it into the surgeries, the doctor's surgeries, is a key thing. And I don't know whether it's the responsibility of the researchers or how it can be done, but that is vital. And then, of course, as Isabelle was saying, for the GPs or the doctors to be able to look forward and say, okay, this is what, you know, what may be ahead, but these are the things that we sort of found are in place that are going to help functioning for as long as possible. And that doesn't happen. And it's almost as if there needs to be a list for doctors to have, one for the diagnosis, for what they should be looking for, and then covering, you know, what they should be doing then. It's guidelines for GPs and guidelines for neurologists, because the message that, I mean, I work with a lot of researchers, and the message I get from the researchers, I kind of go into the doctors thinking, why don't you know any of this stuff? You know, and if you try to talk to them, then they just think you're a smart ass. And you get sick of being treated as if because you can give them information, there's nothing wrong with you. You know, I've got information because I'm so interested in research and so interested in keeping up. So just getting that message across to GPs and specialists of, one, you know, how to make a diagnosis, what you should be looking for, what screeners are out there, And then, you know, what can be done afterwards. And if I can just take the time, there's one further step that I'd like to sort of look at, and that's about the ongoing support. And I think Isabelle mentioned that. I mean, I live alone, unlike most of people with early stage dementia. I'm living in the community. I'm not in residential care. Being alone, I don't have a carer to oversee me. So I've said to my GP, look, I need you, you know, when I come in, just do a bit of a check on me and see how I'm going, because at the moment I have enough insight to recognize when things are going wrong, but when do I know when I'm not noticing? And my GP's response was, look, my job's to manage things that I can make a difference in, and what's the point of me monitoring how you're going, because I can't fix it, which is, you know, just gives me a total sense of hopelessness. How to deal with that. I mean, we know there are lots of things out there that are making a difference. And so, you know, that this knowledge about post-diagnostic support, allied health particularly, and there's quite a lot of work being done into allied health work, and some research being done into other things that help people to keep functioning, which, you know, sort of the music and the art stuff, there's quite a lot done in that. But I was really interested in Professor Crutch's talk this morning, the rare, I have to read it, sorry, because I can't remember it, the rare dementia support group stuff, because I'd love to see more. We don't have much like that over here. There's virtually nothing. I've been involved in actual fact in setting up a number of dementia cafes through Rotary. We've got 8 running at the moment here in the Sydney region. But I'd be really interested in researchers taking an interest in both how this peer support, with, you know, whether it does make a difference. I mean, if you're looking at it from an individual perspective, I see changes in people that are actually participating in these groups. And, you know, whether it's individualized support, that there's a program just coming out through Dementia Australia where there's going to be one-to-one peer support, which I think is fantastic. The peer support groups, some are running online and some face-to-face. I'll certainly be looking into this rare dementia one. But any kind of incentive to maintain social interaction and avoid social isolation, I think I'd love to see the researchers looking at that. That would be where I'd point at the moment, because I think it's such a key factor. We've all recognised how hard it's been for everybody in regards to social isolation over the past couple of years. But that's our life. You know, it's very easy to become socially isolated because of, you know, all the things that we've talked about. I had a question. Professor Irish, I couldn't put in, it wouldn't let me put a question in the thing, but I was really interested whether with the anhedonia, whether people thought we're trying to overcome anhedonia by pushing themselves to do things because they know they should, not mentioning any names, but actually really not enjoying it very much. You know,
and I think it's a double-edged sword. So we know that we've got more people living alone than we ever have. So we've got a changing sort of group of people living in community who are growing older just generally, whether that's with or without dementia. That's sort of a changing landscape. But I think you're exactly right. I always felt that for the group that sort of had anhedonia, that they were the ones that, particularly once they hit the aged care, they were sort of left alone because, well, they were the ones that weren't causing trouble to anyone. So it was never sort of deemed, and I saw that in the sort of, I've come out of working in psychiatry and then moved into aged care. It's that funny, this is what I was trying to think with Prof. Irish's work. It's those symptoms that sometimes fly under the radar, but then you can't force it either. And I think we had such high hopes for the NDIS with respect to personalised care, but yeah, Monica's in particular been leading fantastic work in that space. And I think it's been one of the biggest letdowns of systems with so much potential. But look, I'll move on to Judy next. Thank you for that, Bobby, and Isabelle. So Judy, thinking from it, we've just heard a whole lot of information about clinicians and the role of clinicians. So what do you think researchers can offer to clinicians then? What do you need from us to inform practice?
I think the lovely Isabelle and Bobby have articulated so well to researchers who the expert is in the room. And so no research should be undertaken without the person with the lived experience and their families, their partners. Let's go and do a root cause analysis to all of this. I've heard a lot about doctors not having the right approach, bedside manager, poor terminology. So we need to go back and include dementia in the undergraduate curriculum, not just for doctors, but for nurses, allied health, care workers, everybody. Yeah. People don't know what they don't know. And this is the problem, that people who don't investigate further into dementia care, they're going to remain ignorant and not understanding. So researchers have a responsibility to ensure that that's not the case at all. So one of the best examples of the not knowing of what you don't know is what's happening in the disability sector. So we have this wonderful funding, and I don't speak to anybody that doesn't say, yes, we need this, we need the NDIS, but they their knowledge base and experience with dementia is underwhelming. That needs to be addressed. So researchers, please, listening to the talks this morning, we need also to evaluate the services. So I'd be very interested in evaluation of the NDIS and in outcomes, what the aim of the service is. The other thing I've got on my radar is clinically, I see from the person with the lived experience, but also the family who are living their own experience, this sense of loss of self-efficacy, and they're overwhelmed with it before they even come to our memory clinic. Disease before dementia, the long and winding road, as Isabella articulated, that you get sent to all these different locations with depression, anxiety, anhedonia, and not surprising, because this is a prodrome of the most common cause of subtype of dementia, Alzheimer's disease. So that can present, you know, a decade or more beforehand. Doesn't mean that that's going to lead to dementia, and it's about us as clinicians articulating that to the consumer so that they can take that knowledge and embrace it and go, okay, what is my care plan when I leave the doctor's office? What is the care plan moving forward? That I've got depression, it's a risk factor for developing dementia. And then buy-in from the clinicians with the consumer. Well, tell me your goals, because it's no good giving you this 5-step plan. Yes, you've got to exercise, you look after your heart and socialise, but what is achievable to the person? Because that gets me back to the self-efficacy. If the person doesn't believe they're going to be able to achieve it, they're not going to buy into it. So I would say the loss, the consummate loss in young onset dementia especially, it's like no other. It's a loss of identity as an individual, as an employee, as a parent, a child, a spouse, as a sexual partner. And the symptomology, the symptoms of the dementia, which is a loss of memory, of being able to concentrate, uncharacteristic behaviour change. So these things include language changes. We heard about that this morning with FTD. And conversely, with all of that loss, the person generally can remain in good physical health. They're very strong. They have strengths. So I did a little bit of investigation about self-efficacy, and a fabulous paper by Harris and Keedy back in 2009 describes some themes around loss. And a couple of them were so palpable, they really hit in, and it included an identity as being abandoned. So in the workforce, all of a sudden you've got dementia and you're sitting at home and all your mates are at work. There's no one there. And there was a quote in the paper about, well, my best friend is my TV now. That's just so wrong on so many levels. And the other theme that they brought up was the theme of identity as an individual engaged in living. And I highlighted on my paper, Isabelle, lots of things that you said, but I highlighted the word hope.
Yes.
And then underneath, Bobby mentioned a sense of hopelessness.
Yeah.
Yeah. What are we doing, folks? What are we doing out there? So people hold core values in them that help define who they are. That remains a constant in dementia, and that's what we need to draw out in them. So researchers, please, self-efficacy, very important. Personally, a question I have is how we convey the diagnosis of dementia. This is actually for clinical governance, because in a memory clinic, we know how to do it. But for clinical governance, my question is, how long does it take to tell someone that they have dementia? Well, besides the fact as long as it takes, which is the guidelines in ADNET. But for us here in Adelaide, 45 minutes we allocate. We need to make sure that the person has the opportunity to understand the pathology that's going on. So looking at brain images, looking at the cognitive results, but also looking at the story that the person brings. So their personality, who they are, what their strengths are. We are very fortunate to have two of the best neuropsychologists in Adelaide in our service. And when we use neuropsychology, they're present at the feedback. The nursing model, I'm present at the feedback and the physician. So it's a multidisciplinary team. Please research that and inform clinical governance, the time it takes and how to make a diagnosis. My last one.
Judy, sorry, I'm going to have to switch over because we're supposed to finish in 4 minutes and we haven't heard from the fantastic Jade yet.
Oh, sorry, Jade.
Let alone my wrap up. So Jade, just bringing us around to that, one of the things that we constantly grapple with in young onset dementia is people throw back, yes, but how many people does it really affect? But it's a rare disorder. So how is it that you think we can make that case better, that this is something that's really important for research?
Sure. I think that is the $1 million question in terms of research and attracting research funds. But I think, sitting here and listening to the stories today, I think the importance and the significance is so apparent and it's so clear. So I think as researchers writing those research grants, then we really have to come up with the clear and compelling statements that do communicate this importance. And I think most importantly, we do need to be partnering with people living with younger onset dementia and who are touched by younger onset dementia, including you in all stages of the research. And I think that has to begin at that earliest point of where we're coming up with the research questions, when we are thinking about how we are going to communicate and sell that to the funders. So I think if we know that we are addressing the most pertinent, the most pressing issues, and we can see what they are, it's relating to timeliness of diagnosis, how well we're linking people with post-diagnostic supports, improving access to allied health, looking at things like therapeutic nihilism that we know still exists in the field, and helping people, making sure they don't fall through the cracks of health, disability, aged care services. So I think we know the issues, and I think it is just up to us to really communicate that in powerful ways. So I think the answers are actually very much within our conversation already.
Thank you. And I know, so one of the things I sort of straddle that research and now teaching role. So I teach medical students and I just get them to sit there. I turn the screens off and I say, sit there. I'm going to get you to listen to the voices of people living with dementia. Listen to the way that they actually are experiencing this. Listen to the way that families and supporters experience this. And it's just getting to actually sit back and take on board the life stories, because you don't know if you can't walk it. Our medical students at UNSW are very young. but oftentimes they connect and they send me emails afterwards and just go, oh my goodness, you know, you really made me reflect on my journey with my family. But I think, yeah, taking times for those reflective moments with our doctors when we're training them are really important. But look, I'm going to zoom around. Can I ask very quickly, Bobby and Isabelle, just very briefly, when is it that you like to be brought into the research process? Because I think do you like to be brought in to start to talk about things, or would you prefer to be brought in when there's a bit of a plan in place and for some comments on how that might work? Because we don't want to make it too early if that's overwhelming, but too late because we can't make changes then, meaningful changes.
Yeah, I think it's I think it's easier when there's a bit of a plan, but not too far into the process, because otherwise we're just there to tick off your project. And I've been involved. There was one like a study in mice, and I thought, well, how are we going to make any comments on the benefits of this or make any improvements? But actually sitting there and bouncing ideas off some of the other panelists, we were all coming up with really creative ideas and putting things to the researcher that they hadn't thought of before. So and that you know, that positive feedback loop, that really helps you as the advocate know that you are informing research practice and you are making comments that are really valuable, and that it has been a collaborative approach rather than, you know, here's this project, do you think we should do it? Yes, okay, off you go.
Yeah, I think you're right. It's the rubber stamp we want to get away from.
Yeah.
Okay, I think Sam's come to wrap us up. Bobby, would you like the last word? You can have about 5 last words.
Oh, okay, I was going to answer the last question, but I'll miss that one then. But except I'm different to Isabelle. I want to be in right at the start. I've worked as buddies for people, for researchers, putting in grant applications, and I've helped to word the actual hypotheses, you know, and that's for me. And there's been shifts, you know, they've been on a track, and I've said, but you know, if we look at this perspective, and that for me, that's really important, maybe because I'm a control freak, but it's kind of, I know what we want, and it's what we've all talked about today. We're all saying the same thing. There's nothing that changes, you know. Let's get the diagnosis right. Let's get the message from the diagnosis right, and let's get the ongoing support right. Because yeah, it's not, a diagnosis is not the end. And we need to get that message across. It's the beginning of a new chapter, and we can get out there and continue to live our lives and be active and have meaningful lives and worthwhile lives, and don't take that away from us.
And it's a beautiful message of hope to finish on then, I think.
Yes, exactly. Thank you very much, Bobby, for your last comments, and also to our panelists with their insights and experiences. It's been a lovely, fascinating panel discussion. I'd also like to thank our speakers. So I know Marin is still online, but also Professor Gil and Sebastian Crutch who could be with us. Adrian, who helped moderate, the rest of the YOD-SIG Executive Committee, and the ADRF for supporting us, but also to Dr. Monica Cations, who actually spearheaded and organised this whole special event. So I'd really like to thank Monica so much for her help, and hope we can host more events like this in the future. So thank you very much, everybody. We should be able to get a recording as well and a small survey for your feedback. So thank you very much. Please come and join us, our group. We're on the AdNet website. Thanks very much again. Have a great day.
Bye.
Bye. Thanks everybody on the panel.